In this video, we're going to look at vision. So vision uses the eyeball to pick up light waves. And so we've gone through a lot of these structures in lab. I'm just going to remind you of basically the path of light as it passes into the eyeball. So the path of light will first pass through the cornea, which is a clear outer covering at the front of the eyeball. Then the light will pass through this aqueous humor, which is a watery humor that you might have observed when you dissected the cow eyeball. Then the light will pass through the lens, which is kind of like a clear marble that is actually made out of some elastic tissue so that it can stretch just a little bit. And it's better at stretching and changing shape when you're younger, and then as you get older, it becomes a little bit less malleable, which is why your vision tends to get worse as you age. So the light passes through the lens, which can kind of change the focus of the light. Then the light will pass through this clear vitreous gel. And then finally, the light will hit the retina. And in the retina is where we're going to have the neurons that are able to pick up these photoreceptive, or the photons from the light. Of note, the pupil is a hole created by the iris. So the iris is the pigmented region of the eye that you see when you look at someone's eyes. And when you're looking at their pupil, that dark pupil, what you're actually seeing is their retina, which should always appear very dark black because it's heavily pigmented to prevent those light waves from bouncing around. That black will enable the retina to absorb all of that light and reflect none of it. So in the retina, this is what we're seeing here. If we follow the path of light, this is the very back of the retina. This is the front of the retina. This is the light coming in. So as light comes in, it's going to pass right through all of these cells and be absorbed by the black pigmented cells at the back of the eye. So this pigmented layer prevents light from reflecting back around inside the eye. And then all of these other layers are called the sensory layers of the retina, where this is where you're going to have your photoreceptors. And the first layer we're seeing here, which is kind of the second to most posterior layer of cells, these are what are called the photoreceptive cells. They're called rods and cones. Cones, I'm going to start with cones, starts with the C, and C is for color. Cones give us our color vision. You have a specific cone that responds to specific wavelengths. For example, we as humans have 3 cones. One of our cones is regarded as the blue cone because it will respond to light of the blue spectrum wavelengths, so those wavelengths of light that produce blue. Other cones will respond to wavelengths of light that correspond to green, and still others will respond to wavelengths of light that correspond to red. Other animals may have additional cones which allow them to see in the infrared or the ultraviolet light spectrums, and still other animals will have less cones, so they'll be able to pick up less difference in the types of light waves that they're seeing. So cones allow us to see differences in what wavelengths the light has. In contrast, rods, they could care less what the wavelength is. They just care that there is a wavelength there. They detect any light. And so they're very sensitive to any amount of light. Cones are not very sensitive to any amount of light. They're only going to care about the light waves that actually are within these ranges for blue, green, or red light. And so if the light wave is not hitting any of those wavelengths and is not very strong, they will not respond to it at all. Rods, however, will respond to just any little bit of light. So these give us maybe more of our night vision, where we're able to we're looking for any light. It doesn't matter what wavelength it is. So these guys are scattered in together, and in this picture, I think in blue, it's showing us the rods, and then in this pink color, it's showing us the cones, and it's not saying what color each cone is, but so there's going to be various rods and cones, which are called the photoreceptors. The rods and cones then synapse with, so here we're seeing a layer of synapses. This is where these rods or cones are picking up the photoreceptors the photons, or the energy from the photons, and causing an action potential that causes them to release neurotransmitters to these neurons that they synapse with. And these second neurons that they synapse with are called the bipolar cells. And if you look at them, well, they look like good old bipolar cells like we saw in the olfactory, where they have a dendritic ending, then they have their cell body in the middle, and then they have an axon ending. So these are the bipolar cells. They're kind of a middleman. And in response to when they have an action potential, they will release neurotransmitters here to a third cell type, which is called the ganglion cells. These ganglion cells also look like a type of bipolar shaped cell, even though they're called the ganglion cells. The ganglion cells have these short little dendrites that receive neurotransmitters and possibly cause an action potential to then pass through its axon. And the axons of the ganglion cells will join with other axons of other ganglion cells to make what is going to be the optic nerve. So it's a little bit interesting that the light is actually passing through all of these cells to hit the pigment cells. And then once the light gets absorbed by the pigment cells, we see the receptive pathway kind of working its way back the other way. First, the photoreceptor is triggered by it, then the bipolar cell is triggered by it, and finally the ganglion cell sends that action potential back through that optic nerve. In this picture, we're seeing a retina. We want to try to observe a couple features of the retina. So in this picture of the retina, what's happening is all of those ganglion cells are all sending their axons back, and they're all going to join in one spot. So these are all merging to form what's called the optic nerve. And only the optic nerve leaves out the back of the eyeball. In this picture, we're seeing at this location here, this is where the optic nerve is leaving the eyeball. And you can't see the axons very well, but you can see some of these blood vessels that are kind of coming alongside the axon. So these blood vessels that we see are also going to go through the same. This is a hole in the back of the retina, because if this is the retina, I'm going to kind of draw another picture of the retina up here, kind of a lateral view. If this is the retina and these are the axons from those ganglion cells, they all need to merge in one location, and then you're going to have this optic nerve leave the back of the retina. But there's got to be a hole for this optic nerve and the blood vessels to pass through, to get from basically the inside of the retina towards the outside, where it can eventually get to the brain. So this hole in the back of your retina for the optic nerve to pass does not have, at this particular region, there are no photoreceptive cells, because there's kind of no retina here. It's a hole in the back of your retina. This hole in the back of your retina is a little off center, so it's not straight back. Actually, straight back for the path of light, when you hit straight back, this region is called the fovea centralis. So this region right here, where most of the light coming into your eyeball will hit, is where we can see the best, because at the fovea centralis is where we're going to have lots and lots of cones. So at our fovea centralis, this is where direct light focuses. Cones are concentrated. There's very few rods. This enables us to pick up little details about those light waves, such as what exact wavelength they are. And so we have our best vision when we look directly at something, so that we are focusing the image of whatever we're trying to look at right on our fovea centralis. That's where we see best. And so when we look at things, we tend to turn our eyeballs and look directly at it so we can pick up all those details. And we're able to see it best when we're looking directly at it, because the light is in focus on this photoreceptive area called the fovea centralis, which is in a region called the macula, but that's where we're going to have our best ability to see details because of all those cones. And so that's also kind of pictured here. Just kind of looks like a dark spot on the back, but in the center of the eyeball. Now that hole that we were talking about for where the axons and the optic nerve are leaving the eye, this is at a region called the optic disc, and it's just medial, meaning toward the nose. So over here's the nose. This is medial. and the optic disc is just medial to the center, which is the fovea centralis. So just off-centered is where you're going to have that hole, which is your blind spot. And in lab, we tried to find it by looking, lining up this plus with our fovea centralis, and then watching when we got this little red dot just at the right distance so that this little red dot was focused directly onto this optic disc, and we found that the little red dot disappears. So that's finding our blind spot, or actually trying to focus or to see when the light waves are not able to be detected because they're hitting that specific region of the retina. That's known as our blind spot. It's typically not a problem in everyday life because, for one, our brain fills it in, and two, we move our eyes around a lot, and we rarely, to find the blind spot, we had to have that lined up at just the right spot, and we really kind of had to be paying attention to watch it disappear. Okay, so the optic disc is the area where the optic nerve leaves the back of the retina. Let's see where it goes next. So here's that optic nerve as it leaves the retina. Now, interestingly, within the optic nerve, you're going to have ganglion cells from both the lateral aspect of the retina and the medial aspect of the retina. Both of those are within this optic nerve as it enters into the brain. But then after the optic nerve enters into the brain, these axons are going to split. And the axons that were coming from the medial retina are going to cross over from both sides. So here's the medial retina. Those axons cross over. This is what's called the optic chiasma, which is this crossing over point for these axons. This is where in lab we kind of tried to play with some questions such as, what if you cut right here, and so if you have a pituitary tumor, pressing right here, so interrupting the action potential that's trying to travel from the media retina back into your brain. What would happen is you would lose vision from your medial retinas. Now, when you lose vision from your medial retinas because of a pituitary tumor or because some sort of damage has happened to your optic chiasma, this disrupts vision from the peripheral world or your lateral world. And that's because light waves from the lateral world are going to shine onto the medial retina. So these light waves are shining onto that medial retina. So when we have a pituitary tumor disrupting these axons coming back into the brain through the optic chiasma, we lose our peripheral vision, or the vision to this lateral world, and we start to see this kind of narrowing, or what's called tunnel vision, because we're only seeing kind of the middle of our visual path. So these are the optic nerves entering into the brain. They cross, some of the axon tracks cross at the optic chiasma, others don't. So from the lateral retina, they do not cross. They just continue traveling on back. But ultimately, all of these axons will travel to join with what's called the optic tract within the brain. And that's going to go first to a neuron within the thalamus. So here we're seeing neurons within the thalamus. And then they continue, and they're going to basically make a map of your vision onto the occipital cortex. Or perhaps I should call it the visual cortex, which is part of the occipital lobe of the brain. But here we basically have a map of our retina on the cortex. And I'm slightly concerned that maybe a moment ago I called these different things, so I want to go back through and make sure I hit these words correctly. This is the optic nerve because it's still outside the brain. This is the optic tract because now it's inside the brain in the CNS. So this was still out in the peripheral nervous system. Once that same path of axons, when it's outside the brain, it's called a nerve. When that bundle of axons is inside the brain, it's called a tract. In this picture, we can just follow where each of these cranial nerves actually enters the skull. So if you remember back when we looked at the skull, you remember it had kind of all these little weird foramen, or all these little holes kind of in the inferior aspect of it. And all of those holes were for these cranial nerves to pass through. And so in the last video, we were looking at the olfactory bulb. Well, it had a foramen that it passed through. And now we're seeing the optic nerves have these foramen that they pass through. Because the problem is all of these nerves are out in the periphery. In order for them them to enter the brain, they have to first pass through the skull bone. And so each of the nerves will pass through the skull bone, and after it passes through the skull bone, it enters the brain, and it becomes termed a tract. Also, blood vessels move through these same foramen. So that gives us an idea for vision.